Hey, Hammy here, back with part 2 of section 5.3, in which we are going to look at sexual reproduction and how gametes are made. In particular, the process of meiosis and how that reduces our chromosome number from 2n down to n. Okay, like I said, meiosis, unlike mitosis, where you have a cell that divides into two cells that are genetically identical, they both have the same amount of chromosomes as the mother cell did. Meiosis we often call reduction division, okay, because it produces haploid gametes, which means half as many chromosomes. So we often call it reduction division. Okay, because it's, again, mitosis is just dividing up the DNA in the nucleus. In meiosis, it's going to divide it down from 2n down to n, half as many. Okay, the phases, we actually go through two divisions. So you'll see PMAT, prophase, metaphase, anaphase, telophase, you'll actually see twice. Okay, now if you remember from our lab, prophase is actually the longest stage. Prophase 1 is going to be one of the longer stages in meiosis. Okay, so the first half or first division we call Meiosis One. In prophase 1, same as mitosis, you can see down here the nuclear membrane breaks down, the chromatin condenses into chromosomes. We begin to see the centrioles and these spindle fibers forming. But an interesting thing happens that we call crossing over. Okay, the homologous chromosome, so this pair right here would be a bivalent, okay, or homologous chromosome pair. So in other words, when you go to have offspring or make gametes, you take a chromosome that you might have had from mom and one that you may have had from dad. And these would have similar genes on for eye color. Let's say this had a dominant big G, and maybe this has a little g over here. So they both have a gene for a certain trait, but maybe different dominant recessive. What happens is these homologous chromosomes will scoot up next to each other in the cell, and they'll have what we call synapses, okay, where the inside ones will actually curl over, pieces will break off, so they'll break right here, and look what happens. They actually switch places, okay? So now over here you would have a big G, but now on this chromosome you'd have a recessive allele. Here you'd have a dominant allele, here you'd have a recessive allele. So this really contributes to genetic diversity, because it takes kind of like the cards you got from mom or genes and the cards or genes you got from dad, and it actually shuffles them and kind of breaks them off and switches them around. So when these things are divvied up, you get a good mix of your parents that you're going to pass on to your offspring. So this is crossing over is an important part that you should remember that happens in prophase 1. The rest of the stages we can go through fairly quickly. In metaphase 1, you'll notice that homologous chromosomes, remember metaphases, middle, okay, they line up in the middle of the cell here. But notice instead of all the X-shaped chromosomes all lined up in a row, you have the homologous chromosomes paired up. Okay, so they just have crossed over. So you see some crossing over here, the red tips and blue tips switching. Okay, but now the pairs are going to line up in the middle of the cell. Okay, now what you'll notice will happen in anaphase 1 is that the whole homologous chromosome is being pulled. Okay, the centromeres here in the center, the two sister chromatids are not being split. The whole chromosome is being pulled opposite directions. The two homologous pair, one of each, is being pulled in opposite directions. Then in telophase 1, okay, again, you see cytokinesis begins to happen here. Nuclear membrane begins to reform. The spindles break down, and it looks like you're starting to get two new cells. Now notice we started 2N. Our first cell that we started with had 4 chromosomes. after meiosis one, look how many chromosomes we're down to. 1, 2. Okay, so now we're down to N, which is 2. Okay, now after meiosis one, our cells are haploid. So already different from mitosis is that these cells don't look anything genetically, they're not the same anymore. Okay, they have a different number of chromosomes. Okay, and then instead of going all the way through interphase again, there's just a short break we call interkinesis, in which you get cytokinesis, okay, in which these divide and you form 2 haploid cells, which I just explained over here. Okay, now we go through PMAT again. Okay, so this, we call this meiosis 2 now. Okay, so we go prophase 2, nuclear envelope. We have these 2 daughter cells. It's going to happen in each one of them. The nuclear membrane begins to break down. You're forming your spindles here. The centrioles are moving to opposite poles of the cell. And this will look a little bit more like mitosis did. In metaphase 2, now you get the chromosomes lined up in the center of the cell, a lot like we did in mitosis. But remember, this cell is already N, both of these cells. They're already N, they're already haploid. But we don't want to have 2 copies of a gene within a gamete, a sperm or an egg. And so we need to break this centromere here. So in metaphase 2, we line up the chromosomes here with sister chromatids lined up in the middle of the cell. And guess what happens next? Right, anaphase 2. Okay, the sister chromatids now are separated. So that centromere splits. And these 2 things, the spindle fibers shorten, and now it's pulling the sister chromatids apart towards opposite ends of the cell. And then finally we get to telophase 2 or telophase 2. Okay, the spindles begin to break down. Nucleus starts to reform in each of these daughter cells here. Okay, nuclear envelopes form. You begin to get cytokinesis. Okay, you get the actual splitting of the cytoplasm. Now notice, how many cells are we going to end up with? Right, 1, 2, 3, 4. Okay, so some differences between meiosis and mitosis. Mitosis, you ended up with 2 cells that were genetically identical, diploid, just like their mother cell. Okay, in meiosis, you're going to end up with 4 daughter cells that are haploid, that have half as many chromosomes as the mother cell. Okay, so it's going to form 4 haploid daughter cells. These cells are what are going to produce the gametes or the sex cells, which I'll explain here on the next slide. Okay, the using meiosis to form gametes is called gametogenesis, which makes sense, right? Genesis or formation of gametes. Now, you can also be very specific types. Okay, you can have spermatogenesis, which is the genesis of sperm or making of sperm, and oogenesis or oogenesis, which is the making of the oocyte or the egg. Okay. Now notice, I love this graphic here. This gives us a good representation of how the 2 processes are different when we look at meiosis. Okay, we start with the mother cell in both of them that are 2N, so the primary spermatocyte and the primary oocyte, okay, which are 2N. Now notice after meiosis 1, okay, in the sperm you have a secondary spermatocyte. Okay, so here's your 2 haploid daughter cells after the first division. Over here in oogenesis, look, you get 1 secondary oocyte, and then you get this thing called a polar body. Interesting. Why does it do that? Okay. Look what happens after meiosis 2 in the sperm. Now we have 4 spermatid. Those are like the 4 cells we just looked at on the last slide. Those will mature into the sperm that we recognize with the tail and the little motors, and they're full of sugar, and they're trying to carry this DNA and find the egg cell, right? Notice that the egg cell does not go through meiosis 2. Right here would be meiosis 2. The egg cell does not go through meiosis 2 until it is fertilized by the sperm. Once fertilization occurs, the egg goes through meiosis 2. Notice it makes just a little tiny polar body again. It keeps all the cytoplasm in that final one haploid ootid, okay, or ovum, okay, and then the two nuclei from the sperm and from the egg, so N plus N, will combine to form a zygote. You were all a zygote. Remember I mentioned that in class. You were all one cell at one time, okay? But why does the egg cell make these little polar bodies, okay? Sorry, this sperm is over here. Here's the nucleus of the sperm. These two combine. That first polar body might actually divide again. That's what they were showing over here. Okay, is that first polar body here might actually divide again. So instead of 4 equal haploid cells, you get 3 polar bodies and 1 big kind of egg cell. Okay, this is called conservation. Conservation of cytoplasm. Because it's this egg cell now, the sperm is just bringing the DNA from the father. Okay, the egg cell now is what has to start going through mitosis and producing more and more body cells and start growing. So it's actually really smart because it keeps all of the cell machinery, all those things we learned about in the cell chapter, like Golgi apparatus, endoplasmic reticulum, ribosomes. It keeps all of those in the one egg cell, okay, that's going to become the zygote. So it has a lot of the machinery it needs to start growing. Okay, then the polar bodies here are just discarded. And then you have the one ootid that becomes the ovum or the egg that is fertilized and then becomes a zygote. Smart, right? Okay, so just kind of an overview of how we use meiosis, okay, to form gametes. That's the only place in the human body where meiosis takes place, is just in the testes and the ovaries to make sperm and egg. All the other cells in your body, when you're replacing skin or other things like that, when you're making more body cells, that process is mitosis. But when we need to reduce the chromosome number, okay, in order to make offspring for sexual reproduction, it's called meiosis. Okay, now I know this was kind of a long video. I might go over it here in class. Also, you need to rewatch this or make sure you write down any questions for me that you may have.